Yes, you're fussy. All right. Hi, biology. Welcome to class. I guess the image of the moon was a little too obvious in this one. Most of you guys recognize this is the moon. It's the only moon. This is an orange. This is a round stone. This is a dog. This is close-up of rocks, a mushroom, ice, sea glass, and beer foam. I think it's pretty impressive that a lot of these look like moons, but it's just there to fool you guys. All right. Yeah, the orange does kind of make you think of Mars. Absolutely. So, as always, starting with announcements. We do have a quiz and a homework. Thought the homework should be due Sunday. Usually I'm doing those on Sunday. Anyway, the quiz is due on Sunday. I'm pretty sure the homework is due Sunday. And the project proposal is due Sunday. You can find information about this in the project module. You can also find where to submit your proposal. And I want to remind you again, if I give you a grade of 2, oh shoot, hang on. I just accidentally closed the waiting room instead of letting people in. And I don't know how to get back to, oh, there we go. Okay. If I give you 2 points for your project proposal, you are good to go. Everything is fine. If I give you 1 point, that means that I put a comment there. And you need to go and read the comment. It may just be, hey, that's an interesting topic. Have you considered this related topic or this, narrow portion of your topic? Or, I know a really good source, you might want to check this out. So if you see a score of 1, look at the comments.
If you commented something to tweak it a little bit, do we resubmit it or do we just take your advice and do it on our own?
As long as I say that you're approved, you just take the comment and accept it. But if I say, that topic is too broad, you need to narrow it down, then you'll need to resubmit.
Okay, sounds good.
Yeah. All right, our first exam is coming up soon on February 7th. We are having a review session on Friday, February 4th from 11 to 12. I will put up a notice on Canvas with the link for that special Zoom session. This exam will cover the material from the beginning of the semester through the February 2nd lecture. It will be a mixed bag of multiple choice, true, false, and short answer questions. It will largely be drawn from the terminology and concepts pages. So you want to go back and study those. That's where that's going to cover probably all of the exam. I almost never pull questions that aren't from there. If you haven't heard, remote learning has been extended until at least February 14th. So unfortunately, we got another, what do we got? 2 weeks to do. And that means that exam 1 will be on Canvas. I plan to post it after the review session, probably on February 5th. And it will be due February 7th before midnight. So we're not having lecture on the 7th, which is the day the exam was originally and still is scheduled for. I will be on Zoom during class. So if you're taking the exam and you don't understand a question or you need me to rephrase something, you can pop into Zoom. I will be working on something else. I'm not just going to sit here for a class period. So you may have to speak up and just say, hey, professor, I have a question. I will be at my desk with Zoom on the whole class period. And I have one more announcement. I screwed up and did not record Monday's lecture. So this is really unfair to you because I tell you that a recording is going to be available and you might not take as detailed notes because you know you can go back and see the material again. So I'm going to write up my notes. I'm going to post them to Canvas. Excuse me. And the notes will cover what I remember covering in Monday's lecture in a lot more detail than I would normally write my notes up. And you can use that to study from. Anything from that lecture that's not in the notes is not going to be on a quiz or exam. Questions on any of this? All right. Time to get to it. Last time, we wrapped up by talking about organic molecules and the carbon atom. And now today, we're going to focus on the biological molecules or the macromolecules. There are 4 categories of biomolecules: carbohydrates, lipids, proteins, and nucleic acids. In general, macromolecules are what we call polymers that are made up of monomers. So poly means many, and mono means one. So it's a chain of many of these subunits called monomers. excuse me. This is not always true for lipids. Lipids are different than the other macromolecules. The monomers are going to have very similar structures to each other. There are going to be small differences between the types of monomers. They're going to be connected together, and the size of nucleic acids, proteins, and carbohydrates depends on the number of monomers that are connected. This kind of makes sense. If you look at proteins, amino acids are pretty similar in size, so the number of them is going to determine the size of the protein. So for carbohydrates, the monomers are monosaccharides. These are sometimes called simple sugars. For proteins, these are the amino acids. And for nucleic acid, of course, being DNA and RNA, the monomers are the nucleotides. So we're going to start by talking about carbohydrates. If I'm distracted, the cats have decided once again that the place to play while I'm lecturing is in my office instead of like out in the living room or something. So they're a little distracting right now. Okay, carbohydrates. Carbohydrates have a ratio that is 1 carbon atom for every 2 hydrogen atoms, and there's 1 oxygen atom. So for every carbon atom, there's a single oxygen and 2 hydrogen atoms. That's it for carbohydrates. They don't have nitrogens or phosphorus or other elements in them. That's where the hydrate comes from, because if you look at that, 2 hydrogens and 1 oxygen is a water molecule. The monomer is called a monosaccharide. The polymer is the polysaccharide. You might also see monosaccharides being called simple sugars, polysaccharides being carbohydrates, just using the general term carbohydrate. These are the sugars and the carbohydrates that we eat. So it's the same kind of thing. We need them in our cells for cellular function. Generally speaking, carbohydrates tend to form ring structures. So if you haven't had chemistry, you may not understand this image. So I'm going to walk you through what this is. There's a ring. This carbohydrate is bonded to this carbohydrate, which is bonded to this carbohydrate, bonded to this carbon. What am I saying? Somebody correct me when I do that. This carbon is bonded to this carbon. It's bonded to this carbon, this carbon, this carbon, this oxygen, and back to the first carbon. This carbon is also attached to another carbon that has 2 hydrogens and an oxygen plus a hydrogen attached. And then there's a hydrogen, because carbons really want 4 bonds. Scientifically, carbons are most stable with 4 bonds. The other carbons in the ring are all connected to an oxygen which has a hydrogen attached to it, and a hydrogen. So there's 1, 2, 3, 4 bonds. This is glucose. Glucose is often abbreviated C6H12O6. Glucose is very, very, very important in cellular metabolism. So it's critical for making ATP, which is the major form of cellular energy. So we will talk about glucose a lot. Now, the shape is critical here because there is another monomer that has 6 carbons, 12 hydrogens, and 6 oxygens, but the ring structure on that one only has 5 atoms participating in it. Whereas with glucose, 2, 4, 6, you've got a 6-member or 6-atom ring. The other monomers that we will be talking about are deoxyribose and ribose. These are important for making nucleic acids. So deoxyribose is the D in DNA, and ribose is the R in RNA. They're very, very similar to each other. You can see these are 5-membered rings or 5-atom rings. Here's the difference. Deoxyribose has 1 fewer oxygen than ribose. DE means without or missing. So while DI means 2, DE means without. So it's ribose without an oxygen. Tells you right there in the name. And there's one more important thing I want to mention here. You do not need to memorize structures. So I'm showing you the structures, the shapes of these molecules, but I'm never going to ask you to draw these. If you take a lot of chemistry, you'll get plenty of drawing of molecules. Okay, couple more things before we move on. When we talk about sugar, and you go into your kitchen and you get a bag of sugar down, it is going to consist of monosaccharides and disaccharides. Anybody want to take a guess at what a disaccharide is? Think about what a monosaccharide is and notice the change in the word from mono to di. Two sugars, yes, thank you. 2 sugars, 2 monosaccharides connected together. And so when we think of the sugar that you buy in a packet at the grocery store, it's a disaccharide called sucrose, and there's a milk sugar called lactose, regardless, all of the sugars can be used for quick energy. And when our body is, when our body is converting carbohydrates to energy for the cells, they break the carbohydrates down into sugars. The polysaccharides can be used for medium to long-term energy storage. Plants, we call these starches. In animals, this is a molecule called glycogen. They have different structures, and plants can store energy as a starch longer than we can store energy as glycogen. And it really has to do with the size of the molecules. Fats can store energy in a smaller space than carbohydrates. If we had our long-term energy storage in the form of carbohydrates, we'd be much, much bigger than we are just to store all of those carbohydrates. Polysaccharides are also structural. So cellulose in plants, this is what we call dietary fiber. As animals, we cannot digest cellulose. There are animals that do eat cellulose, like termites, cows, goats. Those depend on a symbiotic microbe to break down the cellulose. And I'm running out of space again. It's not going to let me lift that up anymore. Okay. So when we're talking about animals and fungi, carbohydrate that is structural is called chitin, sometimes pronounced chitin. Chitin is the exoskeleton of insects, crabs, lobsters, other animals like that, invertebrate animals that have that external skeleton. That is made out of chitin. Okay, that is carbohydrates in a nutshell. So now we're going to talk about lipids. Generally speaking, lipids are divided into fats and oils. They are hydrophobic. So they're not soluble in water. They are primarily non-polar covalent bonds in the molecule. Generally speaking, they are mostly carbon and hydrogen. You can look at some of these. This is a fatty acid here, and you can see carbon chain with lots of hydrogens, 2 oxygens at this end. This would be a very short fatty acid. They're normally very, very long with potentially hundreds or thousands of carbon atoms in one fatty acid. When we talk about the types of lipids, I mentioned fats. Fats are used for long-term energy storage in animals, and they're used for insulation. Oils are used for long-term energy storage in plants. So fats tend to be solid at room temperature, and oils tend to be liquid at room temperature. It's not 100% either way. This is just an easy way to kind of guesstimate whether something is an oil or a fat. We'll get into more detail about what makes them solid or liquid in just a second. So both of these categories, these fats and oils that are used for energy storage, are in the form of triglycerides. TRI, that tells you 3. This is a triglyceride. So there's a molecule here, which is glyceride, and then there are 3 fatty acids coming off of it. And again, you don't have to know the structure here. The other common types of fats are phospholipids, steroids, and waxes. We're going to talk about triglycerides here, and then we're going to come back to phospholipids and steroids and waxes on a future slide. All right, so when we talk about, hang on a sec, let me check and see. Yep, there we go. When we talk about triglycerides, and I talk about them having chains of fatty acids, when you talk about dietary fats, you are probably heard the terms saturated and unsaturated, and you've probably heard trans fatty acids. So because these are things that are important dietarily, I want to talk about what these are. A saturated fatty acid have only single bonds between the carbons. Because carbon is most stable, with 4 bonds, that means lots of hydrogens attached. That's why we call it saturated. It is saturated with hydrogen atoms. And these saturated fatty acids are straight. So what this means is that when you've got these fatty acids in a triglyceride, you've got 3 of them, and you've got another triglyceride here, because these fatty acids are straight, they can pack together tightly. That makes them solid at room temperature. And diets high in saturated fats are associated with heart problems. And these saturated fats pretty much only come from animal sources. So animal fats are problematic for heart disease because they are saturated and they pack tightly. All right, how are we doing with this? Am I talking too fast? Okay, I've got a vote that we're good. All right. So, unsaturated fatty acids have at least 1 double bond. They might just have 1 double bond in the whole chain of carbons, but they have 1. And you can see in this unsaturated fatty acid, when you have a double bond, that causes a kink in the fatty acid, which means they can't pack tightly. Therefore, are liquid at room temperature. So again, instead of being all straight, I'm going to have to go this way. You've got a kink in some of them, and they're not going to pack tightly together. There's going to be more space between the fatty acids because of those kinks. We call these oils. And they are common in plants. More common in plants than in animals. So plant fats in the form of oils tend to be better for our cardiovascular system, for our hearts. But there's a catch here. Naturally occurring fatty acids tend to form cis fatty acids. What does this mean? If you've had chemistry before, especially organic chemistry, you know that cis refers to the way this kink is occurring. So cis fatty acids, the carbons on each side of the double bond have the hydrogen on the same side. So you can see in this cis fatty acid right here, the hydrogens are both drawn in the up position. And so there's no hydrogen on the down position for these 2 carbons. For trans fats, carbons on each side of the double bond have hydrogen on opposite sides. So here's a trans fatty acid. One hydrogen looks like it's going up, the other one down. That changes amazingly. That one change changes how our bodies handle those fatty acids. And our bodies do not like trans fatty acids. Trans fats are bad for our health. Now, trans fats are more common in highly processed food. So you have to heat the food to very, very high temperatures in order for a cis fatty acid to turn into a trans fatty acid. High heat converts cis fatty acids to trans. Now, I mentioned that fats are used for long-term energy storage because they take up less space than carbohydrates. They take up less space because they are so hydrophobic, there's not going to be any water around. If we have proteins or carbohydrates, there's enough hydrophilicity or liking water that there is always water associated with carbohydrates and proteins, which means they take up more space. But because these guys are hydrophobic, we can store them in a much smaller space. That's triglycerides. This is phospholipids. Phospholipids are similar to triglycerides, they only have two fatty acids, and they have a phosphate group. So a phosphate group is a phosphorus with 3 or 4 oxygens attached to it. We want to know about phospholipids because they are the major component of cell membranes. So a lot of the things that are characteristic of cell membranes are because of phospholipids. Specifically, what you see here in this blow up, the one end of a phospholipid, what's called the head, this is where the phosphate group is. And that phosphate group is very hydrophilic. And so this end of the this end of the phospholipid likes to be near water. These fatty acids that form this tail structure are hydrophobic. So they don't like to be near water. So if you take a bunch of phospholipids and you put them in water, they will spontaneously form a structure with all the tails facing other tails and the head groups sticking out in the water. In our cells, this is a two-layered or bilayer structure. So the membranes are phospholipid bilayers. BI also means two, like bicycle, two wheels. So this is a simple diagram of a lipid membrane with a phosphate head group sticking out onto both the extracellular space and the interior of the cell, and these tails in the middle of the membrane. This characteristic where the interior of the membrane is hydrophobic helps cells control what crosses the membrane and allows different conditions on either side of the membrane. Things like pH or sugar concentration or certain proteins. So when we talk about cells next week and we talk about some of the organelles inside of the cell, we'll see that the conditions inside those organelles can be very, very different than the conditions just here in the cytosol, the loose interior of the cell. And as an aside, most soaps are made of phospholipids. So when you wash your hands with soap, the soap forms structures with a tail in the interior and the heads on the outside. And that allows hydrophobic dirt and oils from your skin to get trapped away from your skin inside these little structures. And the hydrophilic head group means that they come right off and go with the water that you're washing your hands with. That's not really important for you to know for class, but it's something that's interesting. All right, steroids. Again, you do not need to know these structures. I am not going to require you to know structures. The one on the left is your basic background for lipid steroids. There are also protein steroids, but we're talking about the lipid steroids here. And so that's a stiff structure with 4 fused carbon rings. So this is a carbon ring, but the carbons that are numbered here, 5 and 10, are also part of ring B. And ring B shares carbons 8 and 9 with ring C, which shares carbons 13 and 14 with ring D. And it always has this basic shape to the steroid. 6-membered ring, 6-membered ring, 6-membered ring, 5-membered ring. And they're always connected this way. So the primary difference between the lipid steroids is what else is attached. If you're familiar with chemistry, what functional groups are attached? Functional groups are just a collection of atoms that have common chemistry. So this group, this OH group, that's going to have the same chemistry no matter what it's attached to. And it's very, very common. So rather than having to say an OH group all the time, we call it a hydroxyl group. And when we get to proteins, we'll see there are some more of these very, very common functional groups, and I'll explain them a little more detail at that point. So what's attached to the ring structure matters. And of course, our example is cholesterol, which is very important in membranes. It provides stability and flexibility. Cholesterol is a small but very, very stiff molecule. So it prevents those phospholipids from getting too crowded together and allows for more flexibility in the membrane, as well as for making it physically stronger. The other group, the waxes, I don't have any pictures of these. They are very hydrophobic, and they are used in waterproofing. So if you've got any succulents at home or cacti, and you notice that you'll notice they have a waxy layer on the surface of their stem, leaves, whatever, those are waxes. You also see waxes in beehives. The honeycomb, those hexagonal structures are made of waxes. And that is our exciting adventure through lipids. It is a very incomplete discussion of lipids. There's a lot of diversity in shape and in function, well beyond what we talked about here. But that's like a whole nother class if you wanted to get into that. Okay, proteins. These are polymers of amino acids. This is an amino acid. So it has, here we go, here's another functional group. This is what we call the amino group. It's got a nitrogen, 2 hydrogens attached to the nitrogen. On the other side of a carbon atom is what we call a carboxyl group. A carbon that's double bonded to one oxygen and then has an oxygen and hydrogen off for the other bond. This central carbon also has a hydrogen attached, and then it has a side chain that is designated here as R. Oops. Did not mean to close that. The characteristics of R determines the identity of the amino acid. Oh, I forgot to erase this. So these are some examples of amino acids. What is shown highlighted here is the R group. So the top part is the same for all amino acids. There's your amino, there's your carboxyl, amino, carboxyl, et cetera, et cetera. You can see there's a lot of variety, even if you're not really comfortable with understanding structures. You can see that that R group can be really, really small. Glycine only has a hydrogen there. Or it can be a long chain. It can have ring structures in it. So we can divide up the different amino acids in these different categories: nonpolar, hydrophobic, charged, either positive or negatively charged. So you can see these guys tend to have a charge on a nitrogen atom, or a negative charge, which tends to be on an oxygen atom, or hydrophilic or polar. And I'm going to go back to the previous slide so I have more room to type.
Oh,
Does somebody have a question? Okay. So, functions of proteins. Metabolism in the form of enzymes. And remember, when we're talking about cells, metabolism includes making new molecules and breaking down molecules. And enzymes do those jobs. Proteins are helpful to support things like keratin in hair and nails, and collagen in skin, tendons, and ligaments. Proteins are important in transport, moving things around the body and across cell membranes and within the cell. Proteins function in defense. Your immune system primarily uses proteins. Regulation. Is animal collagen bad for somebody who is mostly vegan? Ooh, that's an interesting question. That was a direct message to me, so I'm not going to out who asked this. I assume you're talking about eating animal collagen, in which case there is no difference between that and other proteins. Collagen and other proteins are all going to be broken down in your digestive system. I have seen products with collagen to put on your skin, and that is just going to get washed off next time you take a shower. It's not going to get into your skin. So is it bad for you? Um, it's an ethical question. It's not doing you. Let me think about how to say this without getting too deep into the weeds here. It really is an ethical question. If you don't want to consume any animal products, then you can get the protein you need from plant products. However, there are some tricks to that. If you are vegan and you don't consume eggs and you don't consume dairy products, you may not be getting all of the amino acids you need from your diet. Our cells cannot make all of the amino acids. So we have to get some from our diet. If we eat animal protein, it's easy. We get it all from our diet. If you're vegetarian, but you eat dairy and eggs, you get all the amino acids you need from your diet. If you're vegan, you have to pay attention to your sources of protein to make sure that you're getting all of the amino acids. So animal collagen is, I wouldn't say it's bad for you in terms of health. But as far as ethically, that's kind of a personal decision. Does that answer your question? It's a really interesting question. Really glad you asked that, because it did allow me to point out that we need amino acids in our diet. We've got a much better understanding of the vitamins and the amino acid mixtures that we need from our diet than we used to. It used to be really, really hard to have a healthy vegan diet because we didn't understand it. It's much easier now. I am going to add the caveat. People with certain digestive issues or health issues cannot get everything they need from a vegan diet. So it can be something as simple as an intolerance to soy. Yeah, there you go. Soy allergy. That makes veganism, like you said, extremely challenging. There are other people who, for whatever reason, are not able to digest protein from vegetable products efficiently enough to get all the amino acids they need, or they may not be able to take up certain minerals from their diet as easily as other people, which make a vegan diet challenging. I have a friend of mine who's recently been told by a nutritionist that she has to add some kind of animal product to her diet because her health had been declining on a vegan diet. She was working with a dietitian to try and figure out the problem. And it just seems to be that she simply cannot absorb or digest something she needs from her diet. I know I have problems with nuts and with soy. It would be really, really hard for me to be a vegan. If you want to be vegan, make sure you do your research and make sure you're getting all of your amino acids. That was an excellent aside. Thank you for that one. Okay. Going back to protein functions. I was starting on regulation. When we were talking about steroids under fats, I mentioned that there are protein hormones. We call these non-steroidal hormones. And these are things like insulin and growth hormones. So they can be very, very important, obviously. And we're animals. Motion is a very important function of proteins. Plants are capable of limited motion. They're not using, to the best of my knowledge, they're not using proteins to do that. But now that I start saying that, I don't think I've ever learned what allows plants to do their limited amount of motion. I think I just discovered a new rabbit hole, something for me to explore this weekend. Yes, I'm a science nerd. Okay. So when we talk about the amino acids, there are 20 common amino acids in animals. There are a few others in different organisms, and there can be modifications to amino acids. So after you make a protein, an amino acid might get modified. The number and type of amino acid determines the shape and therefore function of a protein. Structure and function, those always go together in biology. And as I mentioned, some of these are what we call the essential amino acids. Have to get from our diet. No way around it. And before we start talking about protein structure, I want to make sure that you guys are familiar with some of this terminology. We'll see the term peptide. A peptide is 2 or more amino acids. There's polypeptide, which are longer chains of amino acids. And then there's proteins, which have a 3-dimensional shape. And they tend, the cutoff for most biologists between a polypeptide and a protein is right around the 100 amino acid line. Not everybody agrees on this particular number. There's no firm definition of the difference between a polypeptide and a protein. But proteins are definitely bigger and they have a complex 3-dimensional shape. And polypeptides tend to be smaller than proteins, but bigger than peptides. But there's not nice, neat lines between these categories. So when we are talking about the shape or structure. Structure is just another way of describing the shape of anything in biology. We have different levels where we can describe the structure of a protein. So we have what we call the primary protein structure. And the primary structure is the sequence of amino acids. Proteins consists of amino acids connected, one amino acid connected to the next amino acid, connected to the next amino acid, connected to the next amino acid. It's always a chain when it's being formed. So a primary protein structure would be saying the amino acids, alanine, threonine, glycine, cysteine, another alanine, et cetera, et cetera, et cetera. It's literally the list of the amino acids. Then there is some secondary structure, which is local folding. Folding is just the fancy word to talk about the shape and how the protein gets into its shape. So when we talk about localized folding, we tend to see two major shapes in proteins. One is a helical shape, looks kind of like a corkscrew, and the other is a beta pleated shape, which if you've got a sheet of paper and you fold it kind of like a fan, that is kind of what a beta sheet looks like. And because you can fold the the paper into that shape, that's where we get the word protein folding from. Tertiary structure is the three-dimensional shape of the protein. Proteins come in an incredibly wide variety of shapes. And shape determines function. So some of them are going to be kind of clumpy, some of them are going to be long and stringy. There's a lot of complexity here. There can be small regions of these secondary structures, but the final complex three-dimensional shape is the tertiary structure. There's also quaternary structure. This is when more than one protein binds together to form the functional unit. So you can have two different proteins that bind to each other in order to function. You can have 100 different proteins that bind together for the final structure. All right, I had to hide the chat box for a second. Nothing's showing up in there yet. All right. Oops. Now I mentioned protein folding. This is the process going from primary structure to final tertiary or quaternary structure. Proteins can be misfolded or folded the wrong way. Sometimes these can be repaired. Usually broken down by the cell. In rare cases, they can cause a disease. And misfolded proteins are associated with mad cow disease and Alzheimer's disease, as an example. And one last vocabulary word for the day. Denature. Remember we said without? Denaturing a protein is breaking the 3D structure. So you've got a complex protein and you change the pH or you add heat and the protein denatures. So it is no longer functional. The primary structure is intact. So that strand of amino acids is still a strand, but it's not the right shape anymore. You can see this if you cook eggs. You break open an egg into a pan, and the part that we call the white is kind of clear and it's liquidy. And as you cook it, it becomes solid and white. You've denatured the proteins in that egg. And that is where I want to end for the day. Next week, we'll talk about nucleic acids, and we will move on and start talking about cells. Are there any questions before we all wrap up?
I'm good. Thank
okay.
you for class today.
You're welcome.
Bye-bye.
Bye. Bye, everybody. Have a good weekend.